So, good morning and salam alaikum. I'm very happy to be here. I would like to thank nniw99 for this opportunity to share this session. And I'm going to talk to you about a fascinating field on human milk research. Okay. So, before we move ahead, the basics. Breast milk is the optimal nutrition, the best nutrition for every child. And of course, recommendations state that children should be exclusively breastfed for the first 6 months of life and then continue breastfeeding at least until up to 2 years of age. So when we think about human milk, perhaps we have in mind the picture on the right, that it's a liquid of 87% water, 3 to 5% fat, 1% protein, 7% carbohydrates, basically lactose, mainly, and then some minerals, and with a caloric value of 60 to 75 kilocal per 100 ml. In fact, human milk is more like the picture on the left, a beautiful artistic, I would say, representation of the complexity taken by humanmilk.com, where you see that it's not only nutritive factors that are present there, but also non-nutritive factors like growth factors, anti-inflammatory mediators, hormones, enzymes, antimicrobial factors. So during this session today, I'm going to introduce you a little bit. We will scratch the surface actually on the complexity of human milk and understand a little bit how both maternal and infant factors influence its composition over time. Takes a bit long because it's maybe heavy. Okay, good. So Professor Kolecko already introduced the benefits on optimal growth and subsequent metabolic health for the child. What we also know is that breastfeed and breast milk, I'm repeating breastfeeding and breast milk because I think we should look beyond just the milk composition, but also consider the behavioral component during breastfeeding. I would like to highlight that. It protects against diarrheal disease, respiratory infections, and perhaps respiratory disease like asthma, for example. Then of course, the risk of overweight and obesity, and also shapes the healthy development of flavor and food preferences later in life. Now, what's also interesting is that breastfeeding is beneficial for the mom as well. And this comes from basically observational studies showing that it lowers the risk of breast and ovarian cancer, the risk of later type 2 diabetes and cardiovascular disease, and as well a very important short-term benefit for lactating mothers. There is a connection to potentially lower risk of postpartum depression. Oh, sorry. Okay. So again, Professor Kolecko earlier introduced the complexity of how we look at this type of associations and data. I think the case of risk to overweight and obesity is a very good one. There are numerous studies, mainly observational, some interventional with behavioral interventions that undoubtedly state that breastfeeding reduces the risk of overweight and obesity for children. Now, within these studies have been actually some conflicting results, like what is the impact of duration of breastfeeding? Is exclusivity important? And is there a dose response? Here I show the latest meta-analysis data commissioned by the WHO, where the authors tried to control for the presence of residual confounding in these studies, and the result is that indeed we'll talk about an odds ratio of 0.73. But what does it mean? What could be the mechanisms that can explain actually these observational effects? Is it the link to healthier dietary habits later in life? The regulation of appetite and satiety that breast milk may have a role on? What about the bioactives in milk? like before we heard about protein or hormones, cytokines, and fatty acids. And then I will also not touch upon the important role of gut microbiome. What we need to consider in this area of research, again, I would like to say that mainly coming from observational studies, is the time points, the duration of breastfeeding, the other feeding exposures, and then quite importantly, how do we collect and how do we store the samples that we get. Then data analytics, interpretation are very important, like what can we do about the presence of confounding, reverse causality, or modifiers. And finally, I would really like to stress out as well that the study population is important, meaning we cannot globalize what we see, for example, in European cohorts. We need to think about different ethnicities, the health status, the birth parameters, and the socioeconomic status, which can be actually quite an important factor to understand these effects. So this is just an example to kick off and try to navigate through the complexity of human milk effects. And here I would like to highlight 3 categories. First is the timing, meaning that the variability in human milk composition has maybe an effect based on chronobiology, meaning circadian changes, the feeding occasions, and the stage of lactation. Other group of parameters that influence basically composition and result in this variability is the fact that sometimes we look at and we try to interpret single milk components, where in fact what we see in milk is complex structures. And then the impact of external and internal stimuli, meaning the health status of mother, infant, and the external environment. So my talk will outline studies that emphasize the important role of these 3 categories, and then I will finish on some recommendations and questions for the field for the way forward. Let's go to timing. So timing, what is timing? First of all, timing during one feeding is important. We have the foremilk. This is the first milk that comes out during a letdown. And the high milk. The difference between foremilk and the main difference is, if you see here in the picture, the high milk, which is on the right, is richer in fat. This is basically due to the mechanics of letdown, where the fat globules are the last to be excreted. Then we can speak about timing in terms of daily changes, circadian changes, and then overall the changes over lactation. So some of the circadian changes, compositional, daily changes in milk that have been studied is, for example, in relation to melatonin. This is an old study, but there are earlier studies that actually show very similar patterns, basically measured milk melatonin peaks at night, matching the maternal serum levels. Similarly, cortisol has also described circadian change, where the highest concentration consistently is found in the morning. Again, it follows the maternal plasma circadian variation. And what is astonishing here is that this pattern, this adaptation, basically we speculate the adaptation of the baby close to its mom's rhythms is already present at 1 month of age. If we look at macronutrients and micronutrients, I don't think we can claim there are very strong circadian, let's say, patterns, daily changes. One that has been moderately convincing is on fat. It seems like total fat, triglycerides, cholesterol are higher towards the evening, whereas iron is the opposite. We cannot explain, I think, adequately the one on iron. We don't understand. But in relation to the fat, it could be actually explained by dietary intake or the breast emptying and differences during a daily breast emptying. So again, caution interpreting these results. Let's go to increases over lactation. And this is what the literature predominantly describes, how the milk changes over lactation. One first example is fat content, and here you see actually an internal study from Nestle Research where we have compared total lipids in term and preterm population. And what it is seen, and it's in accordance with other published studies, is that total fat slightly increases during lactation. Caution here that actually fatty acids, for example, fatty acids in human milk are a very well described component that influenced by maternal fatty acid intake. Then another interesting example that I have here on the right is not on total fat, but more species of different lipids. And these are types of sphingolipids, gangliosides, GD3 and GM. And what you see is that whereas the main actually gangliosides in milk, the GD3 is the one that reduces over lactation, whereas GM3 goes up. So again, when we try to interpret these results, we need to understand the dynamics that they have over lactation as a whole, but also within different species. And of course, the very well described, and I will not talk a lot about it because Professor Kolecko also described it before, the protein concentration reduction from colostrum towards mature milk is well described, and nowadays we confidently follow this up, and there are also clinical trials who have tested this hypothesis. Okay, a topic very close to actually my heart. Genetics in general do not seem to influence human milk. There is one beautiful exception, an evolutionary conserved example on human milk oligosaccharides. For reasons that I would love to further investigate. In fact, we know that maternal genetic variation influences the functionality of specific enzymes that produce HMOs, human milk oligosaccharide. The most well-described example is fucosyltransferase 2, FUT2, which produces 2FL. And then in the case of the presence of these genetic variations, common genetic variations, by the way, meaning that they are prevalent in the population, can lead to milk, for example, having complete absence, as you see here in the green bar, of this particular HMO, an intermediate state in case of heterozygosity, and then when you have what we say the wild type in genetics, a full secretion of this HMO. Internally, what we have tried to do, you see on the right, is use genetic variations to try to predict these HMOs without measuring actually milk, but just by maternal genetics. And we constructed what we call a polygenic risk score. Here you see the result on how to predict low and high secretors of 2FL. And in fact, the model shows that the predictability is not too bad. We are able to nicely predict using genetic variants if a mother belongs to a high or a low secretor group. To make it even more complicated, and I think it's interesting, and I'm sorry for the way they are depicted here in the slides. Is that, we spoke about FUT2 gene genetic variation. There is also a second gene called FUT3, which acts in the same way. So if you combine the functionality dependent on genetic variation of these two genes, then you will come up with 4 milk groups, what we call in the population, depending if they have one of the two enzymes functional, both or neither. So I would like to raise your attention in these 4 bars. They are not only different in the type of HMOs that are present in each milk groups. For example, you don't have 2FL in the food to negative people, but also the overall proportions of HMOs are affected. So a totally different HMO profile in each milk group. Then if you add on this dimension the changes over time, what you see on the right, it's clear that when you want to understand better the biology of HMO and any associated benefits, you need to consider milk groups. You need to consider how they change within each of these categories, and then follow them longitudinally per milk group. So what about these complex structures? And I've selected here a few examples. The first one is more on the chemistry side, and it relates to the fat content. So 98% of the milk fat is delivered to the baby in the form of triglycerides. These triglycerides have a very special structure. If you look at the pictures on the left, so what we call the OPO, meaning oleic palmitic acid in the middle, oleic in the third position, is the most common, actually, triglyceride found in human milk. Whereas compared to vegetable oil mix, you have the reverse. The palmitic acid is on the positions 1 and 3. Palmitic acid overall is quite a predominant delivered fatty acid to the baby. Now, why is this structure important? What has been shown is that this palmitic acid structure, and here we talk about structures, not just fatty acids, not just proportions of fatty acids, can influence the nutrient absorption in the gut. How does it do that? Basically, when the palmitic acid is in the middle position, upon the hydrolysis by pancreatic lipases, it can become a monoglycerol, and then the side fatty acids, they are also nicely absorbed through the small intestine wall. In the case of the palmitic acid being on positions 1 and 3, it forms calcium salts. These are, there is a difficulty there in being absorbed. It relates to hard stools, low fatty acid, but also mineral absorption. And in several clinical trials, this has been shown as a reduction of, for example, gut comfort in infants. So the structure plays a role. The way the nutrient is delivered in the infant gut plays a role. Another example I have selected here is the cellular content of human milk. So human milk actually has living cells, mainly lactocytes, immune cells as well, and stem cells. The majority of these breast milk, human milk cells are lactocytes. There are different distinctive, actually, clusters. Some studies say there are 4 or maybe 6. One specific cluster, and this is a very interesting study where they used the single-cell RNA-seq to characterize these specific cell populations. So one particular lactocyte seems to be predominant and increases over lactation. But something interesting that this study also showed is they included the metadata of infant and mother in this study to understand if there is association with the number and the proportion of these cell subsets. What they found is that this particular lactocyte subset seems to be associated with the time, for example, the child spends in the daycare and the presence of hormonal birth control. Another example I chose here is the extracellular vesicles. These are vesicles found in the milk. They are surrounded by single membrane, and they carry protein, lipids, nucleotides. They have been mainly studied, actually, because of their cargo in microRNAs. These are small, non-coding RNAs that have a regulatory function. So here what you see on the right is a study that we've done internally. where we've seen that we have characterized exosomal microRNAs over lactation. And interestingly, we see that there is a core of these molecules, around 35, that remain stable over lactation. So they never change. And some others that are very variable. Most of them actually seem to be associated with regulating pathways on innate and adaptive immunity. So this is an example of a non-nutritive component of milk. And again, why I'm bringing it here is when we study milk as a whole, we need, I think, to integrate not only what nutrition, what nutritive factors are delivered, but how such cargoes here could have an impact on later physiology. Then the last part is the external and internal stimuli, and how does the health status of the mother and the infant actually impact human milk. One very good example is the presence of maternal and/or infant infections. This is a study of 21 mother and infant pairs, where they have characterized the cellular proportion in different stages of lactation. And what you see here is the key messages is that in general, colostrum has a higher cellular content, but in the case, of course, of infection, by the way, either mother or baby, so both can have this impact, you have an increase in a leukocyte count, of course, and almost close to what you see in colostrum. What's interesting is, well, if you lay this data as well over lactation, and these are the lower graphs here, is that, of course, you see the red line shows the measurements in subsets that have taken during presence of infection. But you can actually see the power of considering metadata. This here, this square is a cluster of samples with women that during the time of collection have reported, for example, mastitis. Then one interesting, intriguing, I will say, example is season. We think, how could season maybe impact milk? How we could consider seasonality when we try to interpret this largely observational data. But the first question I would ask, what do we mean by season? On the left is a study in Gambia. In Gambia, I presume many of you know, is they have two main seasons, what they call the dry season and the wet season. Compared to the wet season, the dry season has also a difference, of course, in the weather, but also food availability. So what these researchers here describe is that the total measured HMOs in these mothers during this time of higher food availability, the dry season, is more than compared to the wet season. So this is a setting very specific, with very particular characteristics. And then let's go to one that is more common and not maybe as exciting and more normalized. We tried internally to understand the impact of season in terms of daylight in a multi-center European cohort. So in this study, we have found no impact whatsoever on season and specifically daylight in none of the components of human milk we have found. So again, variability also in the way we describe, the way we design the studies is important in later on interpretation. So what I have described so far is that definitely we have in the literature consistent and well-described milk composition variation over lactation, mainly for macronutrients. We do not understand the totality of these compositional changes, but they likely reflect developmental needs of the growing baby. And definitely what we have here is due to the complexity of studying this interplay between milk, infant, and mom, a lack of maternal and infant recommendations, for example, for staged milk feeding or circadian matched expressed milk, which could be important as highlighted from the studies I showed you. Yes. Then some maybe thoughts that I put on the slides is, I think definitely we should agree that, we need to move away from single nutrients to complexes, structure and delivery, and then understand better, deeper the variability. My last slide here, of course we need to continue the deep characterization of composition, looking as well as non-nutritive factors. An idea would be to start thinking about global reference values, considering both subpopulations and stages, and then, of course, test in maternal interventions. Potential now, maybe opportunities we could put in the discussion is refine nutritional needs for mom and babies, safe and efficacious human milk substitutes, optimized use of donor and banked milk, and most importantly, improved breastfeeding support. Thank you very much.